It essentially is looking at a normal person and the amount of fats in their body. You're looking at about 15, can you see where it says weight? All right, 15,000, all right, grams, that is, of fat in a normal person, about 6,000 protein, and then you have very little glycogen and glucose in the body. So the fat obviously is a lot more, and it provides approximately 6 times more energy if you look at the last column in that table. So fat is the major energy store in the body. It's the major constituent in terms of the nutrient in the body, and it's the major energy store in the body. Now, in what form is this fat? It is in the form, as I told you last week, triglycerides. Okay, and the triglycerides are essentially found, because they are made in the cytoplasm, they're essentially found in the cytoplasm as lipid droplets. When you break down the triglyceride, you're not going to get energy immediately. You're going to get glycerol and then 3 fatty acids. And you know most of this now. The glycerol is used in gluconeogenesis and converted into glucose. The fatty acids are then broken down into acetyl-CoA, and that's what we have to look at today. The diagram on the left-hand side shows you the mobilization of fat, and you notice the fat that's coming from the duodenum, the little arrow that goes from the duodenum, that fat is in the form of chylomicrons. Okay, and once that chylomicron is made, a chylomicron is essentially fatty acids with lipoproteins and phospholipids around them, and lots of cholesterol in the middle of that thing. It looks like the structure on the bottom right-hand corner. You can see the membrane, you can see a little bit of cholesterol, you can see these proteins, and in the middle you find your triglycerides. So that is what a chylomicron will look like. And the chylomicron is made by the enterocyte, flows in the blood, and ends up going to the liver and the adipose tissue. What does the liver do with it? The liver will break down certain fats inside the chylomicron. In other words, it changes the composition, and the liver then packages it into VLDL. Okay, a lipoprotein called VLDL. Again, that would be put into the blood, and that will flow around in blood and be broken down by lipoprotein lipases. I've shown you the generalized structure. Obviously, it's a whole circle, but I just showed you like a little triangle bit of it. Now, if you look at these lipoproteins that are flowing in blood, this is what you will see. Start off with the intestine on the left-hand corner, okay, and you can see your chylomicrons. Just to help you along here, follow the colors. Follow the colors, and the colors tell you that the blue, can you see the blue on the outside? That would be the protein, okay? The yellow on the inside, if you look at the color again, are the triglycerides. And the orangey dot in the middle would be the cholesterol. And each little lipoprotein, if you look at each one of them, has a varying or a different amount of blue, yellow, and orange. Can you see that? So chylomicrons have some protein, a lot of triglyceride, and a little bit of cholesterol. On the other hand, if you look on the top right-hand corner, HDL, can you see HDL? Has lots of protein, very little triglycerides, and a small amount of cholesterol. Can you see that? I'm talking about top right-hand corner where it says HDL. So depending upon the amount of protein, cholesterol, and triglycerides, you have these different kinds of lipoproteins that are flowing in our blood. Now you might ask, why do we have the lipoproteins in the first place? Why don't we just put the fat into the plasma? Because we can't. Simply because the fats will not be soluble in the plasma, and all they will do is they will stick to the blood vessels. So we have to turn them into these, what Dr. Peter will call micelles, lipoprotein micelles. And that's the only way we can transport them. Okay, let's come back to the diagram. So in the intestine, we make the chylomicrons. The chylomicrons go to tissues. Notice it's going in the blood vessel. And then it goes to tissues, and the lipoprotein lipase will break things down inside the chylomicrons. And that changes the constituents of the chylomicrons. Now, it becomes a remnant chylomicron, and that is then transported to the liver. Now, what does the liver do? The liver repackages that chylomicron into VLDL, as you can see in the middle. Okay, as you can see in the middle, the arrow that's coming down, it says VLDL. Again, that is transported all over the body. Okay, and there you can compare the amount of protein, triglycerides, and cholesterol. You notice VLDL has a lot more cholesterol. Again, that is sent to tissues, such as muscle, adipose tissue, the heart, and so on and so forth. When it gets into the capillaries of those tissues, you start to break down stuff inside the lipoprotein, and that is converted into this thing called LDL. Okay? And LDL, which is low density lipoprotein, can again be transported in blood and can be absorbed into tissues, like in the diagram it shows you, into muscle cells, into adipose cells through an LDL receptor. So these are things that are happening with these fats in your body. And this is where you get the notion of good cholesterol, and that would be HDL with high levels of protein, and supposedly bad cholesterol, or LDL with a higher level of cholesterol and a lower level of protein. Okay, so there's these lipoproteins going in blood. So look at the diagram on the right-hand side. You have a capillary, and there's the lipoprotein coming along. Whether it's a chylomicron or a VLDL or an LDL, doesn't matter. So it's flowing along in a capillary, and as it comes close to the endothelium, you have your lipoprotein lipase that's sticking out of the endothelial cell, and the lipoprotein lipase will essentially start to digest the triglycerides inside this chylomicron or LDL or VLDL. Again, that lipoprotein lipase is triggered to start working. I told you in class last week, it's triggered by insulin. Now I am telling you that a protein that is found on the outside of the lipoprotein called ApoC2 will also trigger or promote activation of this lipoprotein lipase. And again, the lipoprotein lipase will digest the triglyceride into fatty acids and glycerol. Those fatty acids and glycerol will then go through the plasma membrane into the cell. Now, there are 3 stages to this triglyceride breaking down. Triglycerides are degraded into fatty acids and glycerol by 3 separate enzymes. And let's look at this in a little bit of detail. Okay? The fatty acids, you first make the fatty acids, then the fatty acids are turned into acetyl-CoA, which I'll come to in a few slides, and then lastly, the fatty acids, the acetyl-CoA can be broken down in the citric acid cycle to give you NADH and 1 molecule of ATP or GTP. So, This is how we break down the triglyceride now. Again, this is the adipose tissue. Okay? Notice in the adipose tissue we have a lipid droplet. Inside the lipid droplet we have a triglyceride. That triglyceride, again, 3 fatty acids and 1 glycerol. The first enzyme that breaks down the first fatty acid out of the triglyceride is something called, if you look at the diagram, it says ATGL, adipose tissue or triglyceride lipase. Okay? And that will give you dye. In other words, you've got 2 fatty acids with a glycerol molecule. Now you have another lipase called hormone-sensitive lipase, which I talked about last week. And hormone-sensitive lipase will convert the dye into mono. Okay? Monoacyl glycerol. The monoacyl glycerol now will be converted through monoglycerol lipase into fatty acid, the third fatty acid, and glycerol. This is why I don't like this room. Okay, so we have 3 lipases now. So 3 lipases sequentially, one after the other, will chop up a tri into di, a di into mono, a mono into glycerol, and the last fatty acid. These enzymes, if you remember what a fat droplet looks like, you have fat, and on the outside you will have water. So these enzymes are working from the outside in. So they are working at the interface of the oil-water interface. What happens to the fatty acids once they are made inside this adipose tissue or inside a liver cell? They are sent into plasma, and then they are transported attached to albumin. Okay? They're transported attached to albumin in the blood to other tissues that might need them. What happens to the glycerol? The glycerol is sent to the liver and turned into glucose. So these enzymes can be regulated. And the diagram I'm using for regulation is, I'm showing you the activation or the regulation of hormone-sensitive lipase. And if you look at hormone-sensitive lipase, in a phosphorylated state, it is active. And what will phosphorylate it? Protein kinase A. What will increase protein kinase A is cyclic AMP. What makes cyclic AMP is adenylate cyclase. What triggers adenylate cyclase to make cyclic AMP is epinephrine and glucagon binding to its hormone receptors. Okay? And that's what will activate the hormone-sensitive lipase. And to remind you, hormone-sensitive lipase is converting diacyl glycerol into monoacyl glycerol. Also to remind you, insulin, on the other hand, when it binds to its receptor, will activate a phosphatase that will take the phosphate out of this hormone-sensitive lipase and thereby preventing it from breaking down the diacyl glycerol. So insulin has the opposite effect. And that's how you will activate hormone-sensitive lipase. Let's look at this in a little bit more detail. And there we go. The slightly orangey looking round structure at the bottom is the fat droplet. That's where you would find the fat. And around that fat droplet, remember the rest of it is the cytoplasm, so that's the water. So you have the fat droplet and around the fat droplet, you have these enzymes. And you notice you have the HSL, that is the hormone-sensitive lipase. But you also have the ATGL on the top left-hand corner, just where it says number 6. That's adipose triglyceride lipase, ATGL. Remember, that's the first stage. It will take the triglyceride and convert it into a diglyceride. At the same time that enzyme is working, okay, you now need to activate hormone-sensitive lipase. How do you activate hormone-sensitive lipase? pKa will put a phosphate on it, but at the same time, pKa is putting a phosphate on a protein called perilipin. That will activate a protein, okay, called CGI, which will activate AT or adipose triglyceride lipase. So by phosphorylating hormone-sensitive lipase and perilipin, you are now going to activate ATGL as well. So ATGL activated will convert TGs into diglycerides. Hormone-sensitive lipase activated will convert diacylglycerol or DGs into monoacylglycerol. And then lastly, right at the bottom, where you see number 8. Can you see number 8? You now convert monoacylglycerol using the monoglycerol lipase into glycerol and fatty acid. Now you go to number 9. You have your fatty acids now. Those fatty acids will be put into the blood, but now you bind them with albumin. And then that albumin-bound fatty acid ends up going to another tissue, binding to its receptor, and then going into that cell. So this is how you're going to break down your triglyceride. So you end up with your fatty acid and the glycerol. Okay, so let's take this a step further. What happens to the glycerol? Well, there we go. The glycerol gets converted into dihydroxyacetone phosphate and then gets converted into glyceraldehyde 3-phosphate. Once it's converted into glyceraldehyde 3-phosphate, you can reverse glycolysis and turn that into glucose. All right? Now let's look at what happens to the fatty acids. We broke the triglyceride, we turned it into a fatty acid. What happens to that? And there we go. Number 1, all these reactions are taking place in a lipid droplet in the cytoplasm. That means the fatty acid is in the cytoplasm. If it's coming from another tissue, like the liver or the adipose tissue, it is going to come into the cytoplasm of the new cell. So the first thing we need to do to that fatty acid, and I'm looking at the left-hand side, number 1, we need to activate the fatty acid. Just like how we activate glucose with UDP, just like how we activate acetyl-CoA with ACP and malonyl-CoA with ACP, we now need to activate the fatty acid by adenylating it. Okay? So step number 1 is we put an AMP onto the fatty acid. So now we have fatty acyl AMP. Now what do we do with it? It's still in the cytoplasm. We then convert it into a fatty acid coenzyme A. In other words, we can start carrying out reactions with this. So we bring in a coenzyme A, or CoASH. We transfer the coenzyme A onto the fatty acid, and we have fatty acyl CoA. Now what do? Still in the cytoplasm, we need to transport it into the mitochondria, because all the enzymes required to break it down are in the mitochondria. To transport it into the mitochondria, I need a molecule called carnitine. Carnitine is a molecule that will transport the fatty acid, the activated or the fatty acyl CoA, into the mitochondria. I am now at number 3 on the right hand side. Okay? So now we are entering the mitochondria. Once you enter the mitochondria, the fatty acid, the carnitine, is removed and is used to bring in more fatty acid. And now I have the fatty acyl CoA again. Now remember, this is a long chain of carbons, all right? And if you remember how it was made adding 2 carbons at a time. Remember that? When we start to break it down, we start to remove 2 carbons at a time. And I am now at inside where it says beta oxidation number 1, number 2, number 3, number 4, inside the little circle. So with a series of 4 reactions, number 1, 2, 3, and 4, we take out 2 carbons from this long chain. So if there were 16 carbons in this fatty acid, through these 4 reactions, we would take out 2 carbons, and right at the bottom, we will end up with a 14-carbon fatty acid, or fatty acyl CoA. Then that will go back up to the top, and then we go through the 4 reactions again. This time we take out another 2 carbons. So we go to 12 carbons, and so on, until we end up with just 2 carbons. And that would be acetyl CoA. And inside that beta oxidation thing, the 4 reactions, the first reaction is a dehydrogenation reaction. That's just a fancy way of saying you will remove hydrogen and electrons from the fatty acid chain. The next one is a hydration reaction. The next one is another dehydrogenation reaction. And then the last one is a cutting reaction, or a thiolytic cleavage reaction. Okay, so let's look at this in a little bit more detail now. So I have my fatty acid. The first thing I had to do, if you remember from the previous slide, I needed to activate it. So there we are. All right, I have my fatty acid where it says R, C double bonded O minus. That will be my 16 carbons. The R is the 16 carbons with the O double bonded minus. I need a molecule of ATP. I need a molecule of coenzyme A. I react the 2, and I now have my fatty acyl CoA. Out of the ATP, I use one molecule, or 1 phosphate, okay, which I now attach the coenzyme A to the fatty acid. I use 1 molecule or 1 of the phosphate bonds to attach the coenzyme A to the fatty acid, and I make my fatty acyl CoA. Now, to prevent a reversal of the reaction, what happens in the cytoplasm is the remaining ADP is quickly hydrolyzed. It's called silent. It's somewhere on the phone that says silent. Some phones say vibrating. Some phones have an off button. Some phones you can even take out the battery. Okay. So what do? I take the ADP and I quickly hydrolyze it. So I prevent the reverse reaction from taking place. Okay? And that is what I am showing you in that second part. So I quickly convert the inorganic phosphate into pyrophosphate, PPI, and I make a little bit of energy and I use that in the reaction. So that this makes this reaction totally irreversible. Okay? Now I've made the fatty acyl group and now I can use this thing. Okay, I'm still in the cytoplasm. Next step, I need to transport that activated fatty acid into the mitochondria. So I need this molecule called carnitine. Okay? So I bring my fatty acyl CoA, I react it with carnitine, I now have fatty acyl carnitine. Now, once you've got fatty acyl carnitine, look at the diagram on the right-hand side. The green enzyme is called long-chain acyl-CoA synthase. That would be the enzyme that did this reaction. Then, I take my fatty acyl-CoA, the bluish purple thing that says CPT1 is carnitine palmitoyl transferase 1, CPT1. Now what is it going to do? It's going to transfer carnitine or fatty acyl carnitine from the cytoplasm through the outer membrane into the intermembrane space. Okay? When it does that, so I have one enzyme, CPT1 transferase. That fatty acid now comes into the intermembrane space. I now have another transporter or a pore, a translocase that brings the acyl carnitine into the matrix of the mitochondria, and then I cut the carnitine out. And that is done by CPT2. So there are two enzymes that are going to do this thing. And this is how I bring in the acyl or the fatty acid into the matrix of the mitochondria. Now, why am I telling you about these two enzymes? Because people have a problem with these enzymes. And there's problem number one. The most frequently occurring medical problem with fatty acid oxidation is a deficiency of CPT2. Let me remind you, CPT2 is the one on the inner mitochondrial membrane that is transferring the fatty acyl carnitine from the intermembrane space into the matrix. And it is cutting the carnitine out and allowing the fatty acid to go in, and it puts the carnitine back out. Now, what kind of symptoms are seen in patients who have a CPT2 deficiency? It is mostly associated with muscles. It's myogenic or myopathic due to the defect in the muscle version of CPT2. It's autosomal recessive. You're doing genetics, so I think you know what that word means. And what are the symptoms? In other words, how would I write up the question for this? There would be muscle weakness, okay, that can go anything from mild to severe. Rhabdomyolysis means there is muscle breakdown, like dying of the muscle cell. Okay? Generally, these become worse when people exercise, intensive exercise. Okay? And obviously, if the person's diet is high in fat, this is going to become even worse. So that's how I will write up the clinical scenario for this particular deficiency. Okay, now that I have the fatty acid inside the matrix, on the right-hand side of this diagram, you can see the fatty acid on the right-hand side. So we have that fatty acid inside the matrix. Step number one. Step number one, if you remember from the little diagram, is dehydrogenation. That's just a fancy way of saying I'm going to remove a pair of electrons and hydrogens. And what do I do with that? I transfer those electrons and hydrogens onto FAD. So I'm making one molecule of FADH2. Next step, I need a molecule of water. Okay, so that the double bond that I made in my first reaction, I can now reduce that double bond or hydrate that double bond. Third reaction, I take out another pair of electrons and hydrogens, but this time I transfer it onto NAD. So I make one molecule of NADH. And then I come down to the bottom where I have a ketone, and now I cut 2 carbons off from the chain to give me an acetyl-CoA on the right-hand side and a fatty acyl-CoA that is 2 carbons less than what I started with. So if I started with 16, I now have 14. And then what do? I transfer the 14 back up and I start all over again. And then I get 12. And then I send the 12 back up and I start all over again. You follow what's happening here? Until I come to the point where I will just have 2, and when I cut it in the middle, I'll have 2 acetyl-CoAs. And those are the 4 reactions that take place during the beta oxidation of a fatty acid in the matrix. And I wrote the reactions down for you on the left-hand side. The first one is a dehydrogenation or an oxidation reduction reaction. The second one is hydration reaction. The third one is a oxidation reduction reaction again, and the last one is a cleavage. Now, do you need to know the names of the enzymes for each of the steps? I will not ask you those. But this is what is important about those enzymes. I want you to look at the first enzyme. Can you see acyl-CoA dehydrogenase family? Can you see the word family? Yes. Well, what do I mean by family? Well, each fatty acid or length of fatty acid has its own acyl-CoA dehydrogenase. So I have a C16 acyl-CoA dehydrogenase. Are you with me? And that will work only on fatty acids that have 16 carbons. Then I have a 14 carbon acyl-CoA dehydrogenase. Then I have a 12 carbon acyl-CoA dehydrogenase. You understand what I'm getting at? And the 12 carbon one will only work on fatty acids that have 12 carbons. It will not work on an 18 carbon fatty acid chain. You follow what I'm saying? And that's what I mean by a family. Now, why am I telling you this? Because people who do not have medium chain acyl dehydrogenase or acyl-CoA dehydrogenase. Now, again, what is medium chain? Well, medium chain will be 8 carbons, 10 carbons. Okay? So people who do not have medium chain acyl-CoA dehydrogenase, they can break down the 16, they can break down the 14, but the minute they come to 10, they got a problem. It comes and it gets stuck. You understand what I'm getting at? Because they don't have that enzyme to take it from 10 to 8, 8 to 6. So, they suffer from a disease. And there's the disease. It's called medium chain acyl-CoA dehydrogenase deficiency, or MCAD deficiency. It's non-ketogenic because you're not making acetyl-CoA, therefore you cannot make ketone bodies, therefore it's non-ketogenic. Hypoglycemia. All right, they will have extreme hypoglycemia. Okay. During fasting, the hypoglycemia will become very profound or exacerbated. You have decreased acetyl-CoA because you cannot go beyond the number 8 or the number 10. What will the patient look like? Well, there we go. Profound fasting hypoglycemia. Very low or absent ketone bodies. Okay? They will have lethargy. They feel tired, like you are feeling right now. They will go into a coma, and if untreated, they will die. Okay? Again, what are these medium chain ones? They are the C8 and the C10 ones. If this person fasts, especially overnight, 11, 12, whatever it is, the situation becomes worse, or the symptoms become worse. How do you treat this? Okay, you treat with frequent feeding, no fats or very low fats, and high carbohydrates. Are we okay so far? Okay, so what did we do? We took out a fatty acid, and pair of electrons and hydrogen on the top. We took out a pair of electrons and hydrogens at the bottom. We made an acetyl-CoA, and we made a shorter version of the fatty acid by 2 carbons shorter in this reaction. So, if you want to look at the amount of energy that is made by a C16 fatty acid. So start on the top left, it says C16, those are my 16 carbons. So I activate this fatty acid, I use carnitine, I send it to the mitochondria, now I start to break it down. First reaction, first set of reactions. I make 1 acetyl-CoA, that's the CC, the 2 carbons, and now I've got 14. Can you see the 14? In that reaction, I make 1 FADH2, 1 NADH2. I send my acetyl-CoA to the citric acid cycle, and that will give me 12 ATPs. How? Because I make 3 NADHs, 1 FAD, that'll give me 11, and 1 GTP, that'll give me 12. Just for the acetyl-CoA. So now I start adding. I got all my FADH through the beta oxidation. I got 7 of them. I put all my NADHs from my beta oxidation. I got 7 of them. I got all my ATPs from the citric acid cycle. and I add the 14, the 21, and the 96. So a C16 fatty acid chain will give me 129 ATP molecules. Does that make sense? Yes? Good. Now, personally, I don't expect you to know how all this happens, but just to keep in mind that you're making significantly more energy from a fatty acid than you would from, say, glucose molecule. Okay. One slide on peroxisomes. Why? Because peroxisomes can convert long-chain fatty acids. Okay? C18, C20, or 20 carbons, 20, 22 carbons. They can start breaking them down. All right? So peroxisomes contain catalase, you know that already. All right? But they can use that catalase to break down long-chain fatty acids. And babies that are born with peroxisome deficiencies or something wrong with the peroxisome will also show symptoms of fatty acid or the inability to break down fatty acids. Okay? And then there is an acyl-CoA dehydrogenase that transfers electrons. When it takes the electron, remember we transferred onto FAD and NAD, remember those? But this particular one transfers the electrons onto oxygen, turns it into H2O2, and then the catalase will break down the H2O2. But at least you're breaking down the long-chain fatty acids. Now, up to now, it has been rather neat in terms of numbers. But are there any odd number fatty acid chains? Yes, there are. So if you think about this, let's say you had a 17 carbon fatty acid chain. You break up, you take down 2, excuse me, you remove 2, you got 15. You remove 2, you got 13, and so on. You'll end up with 5 at the end. You remove 2, what are you left with? 3. You're left with 3. And this is what we call the propionic acid pathway. So for odd number fatty acids, you'll end up with this last 3. You'll turn that into propionic acid. Okay? That propionic acid will then be converted, and I'll come back to the wordy version. There we are. Beta oxidation, you got your last 2, you convert your propionic acid, you convert the propionic acid into propanol-CoA. Propanol-CoA, you then use, okay, you add a carbon dioxide, you turn it into methyl malonyl-CoA. Now that gives you 4 carbons, and you then convert the methyl malonyl-CoA into succinyl-CoA. Where did you hear of succinyl-CoA? And that will enter the citric acid cycle. So odd number fatty acids will enter the citric acid cycle. And the 2 important enzymes are obviously propanol-CoA carboxylase, where you're putting a carbon dioxide onto the propanol-CoA, and methyl malonyl-CoA mutase, which will just convert, it's an isomerase, convert methyl malonyl-CoA into succinyl-CoA. Now, again, this particular step has enzyme deficiencies associated with it, especially methyl malonyl-CoA deficiency, where the patient starts to accumulate propenyl-CoA in their body, and propenyl-CoA, okay, is acidic. All right, so we've dealt with both of those. Good. Now, what happens to the acetyl-CoA if you are making acetyl-CoA after having eaten the 3 Dunkin' Donuts? Because you've got more than enough. You've got more than enough ATP, you've got more than enough NADH, your glucose stores are full, your glycogen stores are full. What's going to happen here? Well, you could, now look at what's going to happen here. Now we are going to make ketone bodies. And again, during fasting, all right, sorry, I confused you a little bit. That was the reverse thing. During fasting, how many of you go on the ketone diet thing? Well, if you're trying the ketone diet and sometimes they tell you you need to fast for 2 days in the week, the idea is this. If you had dinner last night, okay, within half an hour or within an hour, you've used up all of your ATP. Your muscle, if it's working properly, is also using up creatine phosphate. Within 4 hours, 4 to 6 hours, you are using up your glycogen. Within 8 hours, you've gone into gluconeogenesis. All right? That means by the time you come into 12 hours or 14 hours, you are still in gluconeogenesis. All right? But you need to maintain your blood glucose level. Now your body stimulates or releases glucagon and epinephrine. And you're still not eating 12, 14 hours later. The effect of the glucagon and the epinephrine is to start breaking down fats. So, okay, you're using the fats and you're generating acetyl-CoA ATP. Okay? Now, you're making a lot of the acetyl-CoA, and now you are in the 18 hour of the fast, or the 20 hour of the fast. The acetyl-CoA starts being converted into ketone bodies. And ketone bodies are acetone, beta-hydroxybutyrate, and acetoacetate. And the reason you start converting this into ketone bodies is because your brain, because you're not eating any carbohydrates, your brain is asking for glucose. But your brain can also use ketone bodies. So you start to make ketone bodies. And you are into the 16, 18, 20, 22 hour of the fast. And obviously to make the ketone bodies, you're breaking down the fat. So that's where the logic comes about. All right? And that's what I'm describing to you on this slide. So we are going to make ketone bodies when we break down these fats during extreme fasting. So, insufficient, when you are fasting, you're breaking down your fats, you do not have enough things like oxaloacetate, succinyl-CoA, and so on and so forth. So you now start converting the fat, the acetyl-CoA, top right-hand corner, the acetyl-CoA into, you take 2 acetyl-CoA and you then convert it into acetoacetyl-CoA. You're bringing 2 acetyl-CoAs together. The acetoacetyl-CoA through HMG-CoA synthase is converted into HMG-CoA, hydroxymethylglutaryl-CoA. HMG-CoA through the HMG-CoA lyase is converted into acetoacetate, which is your first ketone body. Acetoacetate can be converted into acetone, and acetoacetate can be converted into beta-hydroxybutyrate. Those are put into your bloodstream. It's made in the liver, put into your bloodstream, and they go to muscles and they go to your brain. Now, why does your body make this? Because you have too much of acetoacetate or acetyl-CoA. You need it, you're breaking it down. Thank you. You're making this ketone body. Why does your body make this thing? Because your muscles and your brain has a very special enzyme called, if you look at the top right-hand corner, okay, let me show you the slide. There we are. Okay, so if you look on the left-hand side, the liver makes these acetoacetates and beta-hydroxybutyrates, puts it into the blood, sends it over to the cardiac muscle, sends it over to the brain. The acetoacetate then gets into the cells, the beta-hydroxy gets into the cells, and now that can be converted back into acetyl-CoA using an enzyme called thiophrase, Thiophrase. Thiophrase converts the ketone bodies back into acetyl-CoA. Now, you may ask, well, why doesn't the liver use ketone bodies? Because the liver does not have this enzyme. Only the brain and the muscle cells have these enzymes. Okay? So liver cells are free of thiophrase, therefore they will not be able to use ketone bodies to break down acetoacetate and so on. The problem with ketone bodies is because it is acidic, it will decrease pH in people who are hypoglycemic, and especially in people who are diabetic. Being acidic, you can pick this up in the blood or in the urine, and if a person has high levels of ketone bodies, they end up having acidosis, or diabetic ketoacidosis, DKA, that's what it's called. This is the last summary slide, looking at last week's lecture on the synthesis, and today's lecture on the degradation of the fatty acid. And there we go. Thank you. Let's do the register.